Okay, hello, welcome. Okay, hello, good day, everyone, and welcome to this webinar series. Okay, welcome, Kanav. We have 2053 and the computer. Let's use the chat box to introduce ourselves, your name, and where you are joining the call from. Looks like okay, welcome, Baxi from India. You're welcome. Thanks for joining. Okay, welcome, Balikis from Nigeria. Mr. Timothy Emmanuel from Ghana. Afolabi from Nigeria. You're welcome. Welcome. It looks like we have about 50 participants on the call right now. Okay, like I said earlier, I introduced myself. I'm Abiyakin Ahmed, and I'll be the moderator for this program. And welcome to this month's webinar series from data to discovery. This webinar is hosted by GNatures. GNatures is a subsidiary of Genomark Holdings, and we utilize African medicinal plants and microbes to provide sustainable solutions for global health challenges by integrating genomics, artificial intelligence, and medicinal chemistry. We are committed to pioneering research in translational medicine and natural healing. Okay, what we do in GNatures, we are innovating sustainable health solutions through African medicinal plants and microbes. One of our services in GNatures is translational research and development. We also carry out product creation and commercialization, personalized wellness and consultation, innovation and technology integration, education and community outreach, just like this webinar, and also strategic partnership and collaboration. GeneTours offers a range of natural products, including remedies for diabetes, diarrhea, and hemorrhoids, alongside versatile virgin coconut oil, all crafted to promote holistic health and wellness through nature's power. Okay, how can we help you in GeneTours? We discover the power of natural healing. Our experts are here to guide you through your journey to better health and well-being. Okay, and mentor and a researcher in GeneTours, and she's already on the call. Let's welcome Miss Christina as she joins us, please. Let's just welcome her. Just use the reaction button to welcome her. Thank you.
All right. Thank you so much. Good evening, everyone. Good evening. You are all welcome. Or perhaps, let me not say good evening, let me say good day, because we have quite a lot of participants here today. So, good day, everyone. You are welcome to this wonderful workshop. To this wonderful workshop. So, I can see those from Pakistan. You are all welcome. So once again, my name is Additional Christiana, and I'll be taking today's session. I believe we are all aware that this workshop is a 2 days workshop. So today is going to be the theoretical aspect, and tomorrow we are going to be diving into a hands-on practical session. All right, so I'll be sharing my slide very shortly. While I'm on that, I would like us to tell me about your current level of bioinformatics. How can you gauge your current level of bioinformatics? Are you a beginner? Are you a intermediate? Are you a professional? Or are you just hearing about bioinformatics for the first time? Or you are just an explorer? Can we just do that very shortly while my screen comes up? All right, intermediate, beginner, first time, wonderful. Intermediate. Oh, this is very wonderful. Okay. All right. Intermediate, beginner, first time. So I'm happy about this. I'm happy about this, that this workshop was able to cut across a whole lot of people. So I'm actually happy about that. I'm happy about that. This is wonderful. So I believe my screen is up now, right? I want to believe so, that my screen is up now. Or okay. All right, thank you so much. Thank you so much. So before I start presenting, I just want to say again, just like you know, that this workshop was prepared for our community, was prepared by GeneTours, and this is one of our passion, that we drive bioinformatics, or most especially based on what we GeneTours are into in plant discovery and research. Plant discovery and research. So, and this is why we created this workshop, so that everybody, because when we talk about bioinformatics, it is something that it is the future of life sciences. Yes, it is the future of life sciences, and we want to see that you are well grounded in this. So very shortly, once again, the second thing I would like us to do is that now you've told me this is directly to those that are maybe professionals or intermediates and all of that. I would like you to tell me what bioinformatics tool have you learned before now? That's the second question I would like to ask. That's the second question I would like to ask. What bioinformatics tool or bioinformatics skill are you conversant with? So can we do that very shortly? Can we do that shortly? What tool or skill in bioinformatics are you conversant with? Okay, Linux, Python, Primer Design. Okay, Genius. When you talk about Genius, that's a software tool that can do a whole lot of things. Okay, data analysis using BioPython, Linux. Oh, this is beautiful. Docking analysis. All right. Thank you for that. So I'll be diving on to my presentation now. And what we have here, just like you already know, is what as you already know is that we are dealing with bioinformatics bridge to real world application. Real world application. And our goal here today is very simple. It's actually to help you move beyond tools and start thinking like a researcher, connecting what you've learned in bioinformatics to real scientific discovery. You know, one of the mind behind today's workshop is because we've had many learners come our paths a whole lot of times, and this is some of the challenges that we see that quite a lot of people face. And so as we move into this, we are going to understand better. All right, so we see that in every life science today, you know, we are flooded with data. But when we talk about data, you know, we are dealing with life science, we are dealing with biological data, and those biological data, they are genomes, they are proteins, they are expression profiles, and many more. There are many more. So, and every discovery begins with data. Begins with data. It could be DNA sequences as in the genomes, or it could even be protein sequences, protein structure or gene level expression. When we talk about expression, we talk about RNA data, or even environmental samples. So every discovery begins with data. But yet, the truth is that data alone does not create knowledge. I don't know if we got that. Data alone does not create knowledge. We having data alone does not lead to discovery. You know, we have to be able to connect the data to a meaning. So our ability to be able to convert this data to meaning is true knowledge. And that is where bioinformatics comes in. That is where bioinformatics comes in. It is that bridge that turns scattered data into meaningful discovery. It is that bridge. Yes. So I want you to think about bioinformatics as a three-way partnership, most especially for those people that are just hearing about bioinformatics for the first time, or beginners. I want you to see the word bioinformatics as a three-way partnership something. Bioinformatics talking about biology, talking about computer science, and talking about statistics. I repeat, biology, computer science, statistics. So when you hear the word bioinformatics, what should come to your mind is biology, as well as computer science, and as well as statistics. That's bioinformatics for you. how you can be able to make use of computer science and statistics to gain insights, to gain meaningful information from the biological data, which we have noted to be genomes like the DNA, or protein or RNA, and so on and so forth. And then bioinformatics is not just about the tools, it's about being able to connect them logically, from data to tools and then to biological understanding. That's what a bioinformatics research is about. So okay, yes, so bioinformatics helps us to collect biological data. So in bioinformatics, you learn how you are able to collect biological data, which is DNA, RNA, protein, and metabolites, how you can process and analyze them computationally, as well as derive insights that explain biological functions, diseases, or adaptation. Yes. So now, why from data to discovery? That means that why this workshop, you know, the theme of this workshop is data to discovery. So why just like I noted from the start, that our goal for today's workshop is very simple, is to just help you to be able to move beyond tools and start thinking like a researcher. Because even as you are learning skills and all of that, really want you to have that mind of a researcher, yes. We've come across the fact that many learners know tools but not how they connect in real research. Just like you might have explored some tools and all of that. Maybe you've heard of BLAST. Okay, I didn't see anybody that talked about BLAST in today's class. In today's I didn't see anybody that mentioned BLAST. Of course, we are going to be exploring this BLAST tomorrow. Yeah, we are going to be exploring BLAST tomorrow. Yeah, so you've heard about BLAST. You might have heard about RNA-seq. Yeah, I saw someone talked about docking. Yes, but where do they all fit? Where do they all fit? And the fact that you might have learned some of these things individually, you might have learned them individually. Maybe there was a program that, okay, you wanted to learn this particular skill and you learned it individually. You might have not seen we've seen some people that after going through a workshop or a boot camp or something, they ask questions like, how do I apply this? Sorry. And that's why we are here today. So you must be able to understand how different tools or different skills can link to research questions. So that's why this workshop came up. How you can understand tools, linking them to your research questions, or how you can know what pipeline suits your field, and then how you can move from data handling to scientific discovery. That is why this workshop is here today. And then when you talk about bioinformatics, the core pipeline from sample to discovery is majorly from sample. That's where you get your biological sample, whether you are getting it from a plant or a bacteria or a community or anything. Sample. You get your sample, you sequence your sample. That's, you sequence the biological data from this sample. Either it's the DNA or the RNA or anything, and then you carry out quality control, or you carry out alignment, or you carry out variant expression, annotation, functional insights, and biological discovery. Sorry. So this is one of the core. This is the core pipeline. This is the core pipeline. But different fields in the life science modify this pipeline, but the principle remains the same. Actually, yes, the principle remains the same. Because when you talk about after you've carried out sequencing, you want to align. That means align align, you are talking about being able to join the sequences together to make meaning, or you want to find out variants, maybe mutations and all of that, or you want to annotate. Annotation has to do with you driving biological meaning to the genes that are present in that sequence, and then you want to drive insight and all of that. So this is one of the core pipeline of bioinformatics. And then across life science fields, this is a typical workflow, maybe in genomics. Now, when we talk about bioinformatics has to do with, we have different fields. Yes, we have different fields in bioinformatics, which is genomics. We have genomics, we have transcriptomics, we have proteomics, we have metagenomics, we have epigenomics, structural bioinformatics, conservation genomics, and so they are so wide, there are so many. The field of bioinformatics is very broad, very, very broad. So if you are dealing with the field of genomics, the typical workflow in genomics is that, of course, everything starts with your sample collection. And then after the sample, you want to get the whole genome sequence. That's what WGS mean. So in genomics, you are sequencing the whole genome. That is everything that makes up that particular organism. You are sequencing it, and that is what we call whole genome. And then you want to align them. You want to carry out alignment. You want to carry out genome alignment, because when you sequence a sample, maybe you sequence a whole genome. When we sequence a whole genome, the sequencing machine actually gives us in it can be fragmented. Yes, it can be fragmented. So we need to align those fragmentations together for us to make meaning of the entire picture of that organism. And that's what we do in alignment. So there are different bioinformatics tools that you make use of to align a sample. I think when I was asking about different tools that you've made use of, or different skills, someone made mention of Genius software. Genius software is one of those softwares we make use of in bioinformatics that can carry out different skills. Yeah, it is a software that has many tools embedded in it. So you can carry out alignments with that Genius software. You can carry out variant calling with that Genius software. You can carry out annotation. So when you're in the field of genomics, what's your goal, what you aim to achieve is actually that maybe you want to identify mutations. Mutations has to do with SNPs. That's what we call single nucleotide polymorphisms, or you want to identify structural variants that affect the structure of that particular, maybe the protein, because when we talk about structure, we are dealing with protein. You want to understand structural variants, or perhaps you even want to explore evolutionary diversity. So if you have a goal towards any of these, that's the field of bioinformatics that answers that question is actually genomics. Or when you are dealing with transcriptomics, the typical workflow is that you get your sample, maybe you get your plant sample, and then you carry out a sequencing called RNA sequencing. In genomics, you are carrying out whole genome. Whole genome sequencing has to do with everything. That's the entire DNA, the entire structure. But in transcriptomics, you are specifically sequencing the RNA sequence from that sample. And of course, you are going to carry out alignment. You are going to carry out expression analysis, expression profiling. Expression profiling has to talk about, because when you talk about RNA data, when there is a genome present, when there is a gene that is present, when there is a gene present, that gene will actually express itself. that gene will express itself, and that is RNA. So that's what we do in want to understand the expression profile of a particular gene. I want to, of course, understand it. Then you carry out pathway analysis. So when you are carrying out transcriptomics, you want to understand the gene regulation, how that gene is being regulated. Maybe at this particular time, let me cite an example. Maybe the way a particular gene, maybe gene A, expresses itself in the rainy season might not be the same way that particular gene is expressing itself in the dry season. So that's what it means. Maybe you want to understand what is happening in different conditions, how that particular gene is expressing itself in different conditions and all of that. So that is what you do in transcriptomics. So please, are we still together? I hope I'm not too fast. Are we still together? Are you following me? Are you following me? Are we still together? I hope you are getting me clearly. Did you say there is no audio? I'm just saying, someone said there is no audio. Is that true? Is that true? Okay, you can hear me. Probably it's from your end. Thank you. Thank you. Thank you so much. All right. So I can continue my presentation. So I was saying, so when you are dealing with perhaps maybe your goal of discovery is that you want to identify key proteins, interaction between different proteins, or post-translational modifications and all of that. You go into the field of proteomics. Or you want to study microbial community structure, the structure of a certain community or function or ecology. You want to go into the field of metagenomics. So when you talk about community, microbial community, that community can be in a particular soil. Maybe you want to explore the microorganisms that are present in a particular community, maybe the soil, in a particular soil, or maybe the roots of a particular plant. What are the microorganisms that are present in that soil? Or it can even be the human gut. I want to believe that we know that the human gut, that is the human digestive system, is actually a community of microorganisms. So you might want to understand what are the microorganisms that are present in a particular individual. What you do there is metagenomics. And the type of sequencing that you carry out is what we call environmental DNA. This kind of sequencing is different from maybe WGS or RNA-seq, because the sequencing that you do in metagenomics, it's going to give you the DNA profiling of many different microorganisms like bacteria, fungi, and so on and so forth. Even in bacteria, you talk about different species of bacteria. But in whole genome sequencing, if you are focusing on just maybe Bacillus subtilis, for example, it will give you just the DNA of Bacillus subtilis alone. Or if you are focusing on the genome sequence of a rice plant, it will give you the entire genome of that rice plant. But when you talk about metagenomics, maybe you want to sequence the bacterial community that is present on a particular rice plant. It will sequence different organisms for you, and that is the eDNA sequencing, metagenomics. That's what you do in metagenomics when you want to deal with a community. Or you want to deal with discovery, regulatory regions, DNA methylation, histone modifications, and all of that. So that aspect of bioinformatics is epigenomics. Or when you are dealing with when you want to predict molecular binding, maybe how or drug protein interaction, maybe how a particular drug functions binds to a protein. You want to study all of those interaction. What you actually, that's the field of structural bioinformatics. So you are going to sequence the protein sequence. I'm going to get the protein sequence. You carry out structural prediction, or you carry out molecular docking and all of that and all of that. So that's just it. That's just it. Or you want to deal with conservation genomics. That means you want to assess genetic diversity of a community, or you want to understand how a plant community is adapting, or you want to even understand the health, because as evolution happens over time, evolution happens time and time and time again. The way something was in the last 5 years is not the same way it is. So perhaps you want to understand evolution of a certain community. You want to understand the evolution. And in terms of evolution, of course, in evolution, mutation happens. And some mutations can be healthy, some mutations can be healthy, and some can be do I say harmful. So when you want to understand the evolutionary health of a particular population, whether the evolution that is happening, maybe the mutation that is happening, is it beneficial to that community or is it harmful to that community? What we do there is for you to carry out conservation genomics. And the kind of sequencing is what we call RAD sequencing. RAD sequencing is a sequencing that is quite different from this other sequencing, because it's a more affordable sequencing, because when you're talking about a population, you are going to sequence a whole lot of samples, and this RAD sequencing is a cheap kind of sequencing technique that helps you to sequence thousands of samples. And then of course you carry out SNP analysis. SNP analysis is that you want to understand the variation that has been happening over time. You want to understand the SN, that's the single nucleotide polymorphisms that has been happening over time. Then you want to now explore the population structure and all of that. Now we can see that in all of this, I think I talked about the fact that bioinformatics helps us to collect biological data. So depending on your goal of what you want to drive out, we determine which type of data you want to collect. And after collection in bioinformatics, you process and you analyze. That's where you talk about maybe SNP analysis, you talk about molecular docking and all of those things. And then from the analysis, you will now be able to now derive your insight, explaining biological functions, diseases, and all of that. So that is how the pipeline goes across different fields of bioinformatics. So now you might now want to ask yourself how to know what pipeline you want to follow. Because like I said again, perhaps maybe you might have registered for a course or something, and you learned a skill. But when we are now dealing with research, like really finding out meaningful research, just pipeline alone or a tool alone does not give birth to a scientific discovery. Instead, it's going to be a scientific question will determine the pipeline or the tool that you're going to make use of. I don't know, do we get that? One of the frustrations that people encounter is that maybe, oh, I've paid for this course, I've learned this tool, but how do I apply this tool? Of course, it's very good that you pay when you have the opportunity, you learn different skills, you learn different pipeline and all of that, because of a truth. When you talk about real scientific discovery, it's going to be a compendium of different tools. It's going to be a compendium of different pipelines. But now, when you have, maybe you've learned a tool in isolation and all of that, and then you are now wondering how you can make use of it to discover, to make a scientific discovery. And then sometimes we see that that's where the challenge comes in. But now, before tools, before pipeline, before you know what to follow, the first question that you're going to ask yourself is, what biological problem do I want to solve? What biological problem do you aim to solve? That's the first thing that settles anything. Most especially for my beginners here. Most especially for my beginners here today, you want to venture into the field of bioinformatics. You want to ask yourself, what biological problem do I want to solve? What biological problem do you want to solve in whatever field that you are? Maybe you are perhaps maybe you are in, you might be a farmer, you might study pharmacology, you might study this, you might study anything you study. Even in that field that you are, what biological problem do you aim to solve? What was the problem in that field that you've been able to discover that you want to solve? That's the first thing before diving into maybe bioinformatics pipeline and all of that, or your understanding of the biological problem that you want to solve will help you to combine tools that you've learned to now drive meaningful information or meaningful discovery. So after you have settled the biological problem that you want to solve, the next question will now be that, what organism, what system, what data set best represents it? You found the problem. What organism will best answer that question? What organism? Is it going to be from plants? That's organism. Is it going to be from microorganisms? Maybe bacteria, or is it fungi that will give you the answer you are looking for? That's the next thing. And then when you settle the organism, the next thing is now, what kind of data can answer it? You know, when we started, I talked about the fact that when you talk about biological data, you are dealing with either DNA, RNA, protein, metabolites, and so on and so forth. So this your problem. Is it a DNA? Is it the DNA that will give you the answer? Is it RNA that will give you the answer? Is it protein that will give you the answer? Or is it a metabolite that will give you the answer? And then the last thing is now that, what analysis connects that data to insights? So you've understood the biological problem, you've understood the organism that can give you that answer. Even from that organism, you've identified that maybe it's DNA or RNA or protein that will give you that answer. Is now going to be what analysis? What analysis? If you are working with a DNA sample, the analysis you will carry out might not be entirely the same with that you carry out when you are working with RNA sample, just like we noted here. So this is it: DNA, RNA, protein, environmental sequence that has to do with metagenomics. Can you see that the workflow, the pipeline among them, there are some pipeline, there are some workflow that are different. Some might share the same and some might be different. So that's it. Okay, let me are we following? So I think I would like to hear response again to be very sure that we are all following through. Okay. Thank you so much. Of course, if you need the mail, we can share the mail. Thank you so much. So let me move on. So okay, so now the lesson from here is that don't start with tool. don't start with the tool. First of all, start with the question. The question is what solves every problem, actually. So you start with the question. The question you are aiming to solve will determine the tool or the workflow or the analysis that you are going to carry out. So that's it. That's it. That's it. All right. So now this is the framework from question to discovery. So you want to define your problem. What am I investigating? Example, perhaps maybe this is what I want to do. What am I investigating? I want to investigate why one plant is drought tolerant. Why is it that this particular plant is tolerating drought and this other plant is not tolerating drought? Can you see? That's one thing about the mind of a researcher. Every real researcher is actually a person that is curious. Yes, you see, curiosity is one of the core skills of a researcher. Curiosity, because your curiosity opens you to biological problems, and then at the end of the day leads you to groundbreaking discovery. So I've noticed that this plant is drought tolerant than this one. So that's what I've seen. And what am I expecting from this, my question? The answer I'm expecting to get at the end of the day is that means I'm going to discover genes that are regulating stress response. So that means, of course, I want to understand what are the genes that are present in this drought tolerant plant that is not present in the other. So when I want to commence my journey of bioinformatics, so to say, I'm aiming to be able to identify the genes that are regulating what? Stress response. Okay. Now, the next thing is that I want to choose data. So what data answers this question? What data answers this question? And a suitable data that answers this question is actually the RNA expression data. The RNA expression data. Because when you look at this plant, there is something that is being expressed. There is a gene that is being expressed in this drought-tolerant plant. That either there are two things. Either the gene is not present in the other plant, or the gene is there, but it is not being expressed. Even from what I'm saying now, I've even diverted into two different fields. Because when you want to talk about this gene is present in this one, this gene is not present in this one, that's the skill of genomics. That's the skill of genomics. You seeing that, okay, in this particular plant, all these, you are able to identify that all these genes are in this plant. And in this plant, maybe this particular plant does not have that gene. It's genomics that will give you that answer. But beyond you seeing what is present, this gene is present here, this gene is not present here. Beyond that is now the RNA expression data that will tell you how this particular plant, how this particular gene that is present is being what? Expressed. So your insight here is the expression profile. It might even be that the two plants, they have the gene, but in drought, something happened that in this one, that gene is not silent. It is being expressed. And maybe in the other gene, it is silent. It is silent. Okay, so that's that. And then after you have chosen your data type, it is now time to select the pipeline. What analysis will fit this thing that I want to do? Of course, RNA sequencing, you sequence your RNA data, and then you carry out differential expression. Differential expression will now help you to identify stress genes, because maybe when you get a plant that you have seen that this plant is drought tolerant, you are going to see some of those genes that are expressed. And another thing, again, even in this expression that we are talking about, it might not just be one gene alone that is responsible for for the drought tolerance. It might be a pathway that this group of genes are responsible. So maybe now you've identified the stress genes that are responsible for this stress. What if in the other one that is not plant tolerant, one of those genes was not in that pathway is missing? So there are a lot of things that you can discover from that, and then you interpret your results. Okay, what does it mean now? After you've carried out all of this analysis and all of that, you've identified the genes that were expressed in this stress condition. You've identified it. Maybe, for example, you identified that it is the ABA pathway genes. Now, I mentioned something about pathway because in a pathway, there are many genes that are responsible. So in this drought tolerant plants that you've sequenced, you've seen that the ABA pathway genes are the ones that are upregulated. So you are now able to discover that, oh, the drought response mechanism of a plant is that during drought, this pathway is much more what expressed. So maybe when it is not in drought, this pathway might be less expressed. So you've been able to discover that, oh, okay, it is the ABA pathway genes. So genes that are found in this ABA pathway are the ones that are regulated. So you've been able to discover like that. So this is just one example out of the many things that you can do. So that's that. So now I prepared a whole lot of different case studies so that it can cut across different fields, because I know that there are different fields that are well represented here today. So let's look at this case study. I want to decoding flood tolerance in rice. So I want to decode flood tolerance in rice. And this field of bioinformatics is what we call plant genomics. Plant genomics. So this was my research question. Why do some rice varieties survive flooding? Why? Why? Some rice varieties. You know that we have different varieties of rice. So there are some particular varieties that survive flooding more than the others. So that's my question. Why do some rice varieties survive flooding than the other? And then the data that I need is that whole genome sequencing, whole genome resequencing, or whole genome sequencing. Whole genome is the entire, that's WGS. So maybe you want to go over to a sequencer where you are going to carry out your sequencing. They ask you, what type of sequencing do you want to carry out? You tell them WGS, whole genome sequencing. That means they are going to sequence everything about that particular rice variety. The whole genome, you are going to see the genes, even the RNA is even part of it. The you are going to see the exon, the introns, the coding sequence, everything is what is in the whole genome sequence. And then the pipeline, you carry out QC. QC is quality control. And then you carry out alignment. Like I said, from sequencing, because the whole genome of a sample of an organism, it used to be very large, over millions of base pairs. Over millions of base pairs. And depending on the sequencing machine, it can only read from the first base pair to the next 200 base pairs. So it will read and stop from 201 to another point. It will read and stop. So it will be in fragments. That's, we call it contigs. And then you have to do what? Carry out alignment. That we do what? Align the whole fragmented sequences so that it can now give you the full picture. So like I always say, when you talk about alignment, it's just like a puzzle. You know, the pieces of a puzzle, when it is scattered, you have to align them together to give you the full picture of what that puzzle is about. So that's just basically what alignment means. And then you've aligned it. You now want to carry out variant calling. Variant calling is a bioinformatics skills on its own. So that means you need the skill of what? Quality control. You need the skill of genome alignment. You need the skill of what? variant analysis, and then you also need the skill of comparative genomics for you to be able to do what? Answer this your research question. So that's it. That's it. Maybe you now, of course, you are going to sequence the different genomes of the rice variety. And based on this question, is a question of why is this one different from this one? That's variant. Variation. What is different in this variety and what is different in that variety? So you need the skill of what? Variant analysis. And then you now do what? Compare. So that's the pipeline you need to carry out when you want to answer this. And then you now discover that after all of this, your analysis, you discover that you identified that SUB1A gene is controlling what? Submergence tolerance. So the gene that is present in this particular rice variety that is helping it to be tolerant to flood is the SUB1A gene, and is not present in the other varieties of rice. So you've discovered that. You discovered that, right? So maybe one of the application is now that, okay, a SUB1 rice is now used, a SUB1 rice is now developed to now used in globally in food pro farms. So this kind of information can guide what? Breeding. How can we breed rice variety that has this gene that will help flood tolerance? And that's it. So in this case, we learned that you started from a trait or a challenge. The trait here was that you saw that this variety is tolerating flood more than this other one. So that was a trait, or we can say a challenge. And then you choose your DNA data, and then you apply comparative genomics. That's case study 1. Let's move to another case study. Another case study is that we want to see microbes that power compost degradation. In this case, we are dealing with microbial metagenomics. And you remember from the start that metagenomics is the aspect that deals with community. So we want to see the microbes. What are the best microbes that do what? Power compost degradation. So the research question is that which microbe make compost decomposing faster? So when we talk about decomposing compost, decomposition, which microorganisms helps decomposition faster? So what do you do? The kind of data here is the 16S RNA sequencing of compost samples. So you get different compost samples at different stages of decomposition. So you sequence, you now carry out the 16S RNA of then of course in that you are going to find different microorganisms, right? So the pipeline is that you have to carry out quality control. Quality control is actually it has to do with, because the data that is gotten from your sequencing machine, sometimes it might have maybe some errors or some gaps and all of that. So quality control is actually what helps us to, is that skill that you have to carry out, or let me say that analysis that you have to carry out to make sure that your sequence, your data sequence is of great quality. And there is in our tomorrow's class, we are going to do a mini version of this, how we are going to be editing and all of that. So be very prepared for tomorrow, because you are going to see some of these things. Okay, so and then we carry out OUT clustering. You want to carry out taxonomy. Why do you need to carry out taxonomy? Because you want to understand because in this sequencing there are going to be different microorganisms. So of course you need to carry out a taxonomy study for them to know which it is at the point of taxonomy that you know that this microorganism is the one that is present here. And then you carry out functional annotation, and then you now discover that Bacillus and Clostridium, they dominate early what? Decomposition stages. So you discover that these are the two bacteria that dominate. And then what's the application? It will now be informed microbial formulations for sustainable composting. So that's it. So in this pipeline now, you've made use of up to how many tools and how many skills like that. So you want to learn taxonomy. You want to learn taxonomy. Maybe when you're talking about evolution, because when you talk about taxonomy, you are dealing with evolution, right? So that's you want to carry out evolutionary analysis. Evolutionary analysis. It is at the point of evolutionary analysis that we know that, oh, Bacillus is present, Clostridium is present, and so on and so forth. And so on and so forth. So that's it. And then you've been able to apply it that, okay, when we are formulating, when we are carrying out microbial formulations, we need to, this bacteria has to be present that will help for sustainable composting. Or if I want to use this this same workflow for maybe you are in the field of nutrition, or you are in the field of let's say, yes, nutrition, or even the medical field. Even this microbial metagenomics can still be applied, because in this case it's going to be the bacterial community. It's going to be the bacterial community of a particular patient, because when you're talking about nutrition, maybe you want to carry out a personalized treatment for an individual. How you need to carry out personalized treatment is that you understand the genetic profile of individual A, the genetic profile of individual B. And one of the truths is that many of the diseases that we actually fight, that we combat, it starts from our gut. Yes, it starts from our stomach. Because when the bad bacteria is overpowering the good bacteria, that's when we now have challenges with all of these diseases. So, or you might want to say, maybe you are in the field of pharmacology, and then why is this drug working for this person and it's not working for this person? So you sequence the bacterial community of this person that this particular drug is working for, and you sequence the other person that the drug is not working for, and then you're able to see the bacterial community. Maybe there's a particular bacteria that is missing in this individual that is not missing in the other individual, and then you now know how you are going to say, okay, this drug works for you and this drug does not work for you. So that's it. It's cut across a whole lot of things. So in this place, we saw that we started from a process. You make use of metagenomic profiling, and then you now discover the functional players. The functional players here are the Bacillus and the Clostridium. And that's it. Okay. Another case study is genetic drivers of cancer drug response. Okay, I think I've also included that in my previous explanation. In this case, we are dealing with human health genomics. The research question is that, why do only some patients respond to breast cancer drugs? So some patients respond positively to some breast cancer drugs, and some respond negatively. Why? Why? The answer lies in the genome. The answer lies in the genetic level, and that's the reason why bioinformatics is actually taking over the life science field, because all of this why, you cannot see it in the physics. You can't see it in the surface. Let me say, you can't see it in the phenotype. You have to understand what is causing it in the genetic level. And that's why, for you to really make impact in whatever field of life science you need, bioinformatics is a very is a field to dive into. Is a field to dive into. All right. So now you now get the data that you're going to get. You get a breast cancer patient tumor versus the normal a normal individual that does not have cancer. And then you now do what? Of course, you carry out QC, you carry out alignment, and then you carry out variant, because of course, in this case, we are dealing with what? Differences between the breast cancer patient and the normal patient. That's variant calling. And then you annotate. Annotation means that you are labeling. I call it labeling in a simple term, because when you carry out the sequencing, the alignment, you carry out variant calling, you are going to see different genes that are present. It is at the point of annotation that you know what each gene, the function of each gene. That is what annotation is about. You being able to understand the function of the genes that you've identified, and then you link to clinical databases and all of that. So the discovery here was that the BRCA1 gene and this particular gene, that it found variations in these genes. Your discovery found out that between the diseased patient and the normal patient, you found variations in this gene. You found variations in this gene. So that means that there is a mutation that might have occurred in a particular individual, and that mutation is affecting how the person is responding to this drug. And that's why we see that when we talk about the medicine has to be revolutionized. We have to come to the point of precision medicine, because as the popular saying goes, what is good for the goose may not be good for the gander. And then because, oh, this person has cancer, then you give the person this drug. Then what if the thing is not working for, it might be working for this person, does not mean it to work for the other person. And that's why we need bioinformatics. We need genomics to be able to really personalize treatment for each individual, because that's just the truth. It's not every every drug that works the same way for each individual. And it's as a result of the genome. I think some months ago we had a boot camp where we for nutrigenomics, talking about personalized nutrition. I don't know if people that attended that boot camp are currently on this call, are currently in this workshop today. You know, because of course you have to, maybe when you're in the field of nutrition and then you want to personalize a particular food for a person, how you are going to know is when you actually, that's from the point of microbial metagenomics. When you understand the microbial composition of individual A and individual B, we guide your personalization of nutrition for such person. So in this case, in the case of clinical now, maybe for those in the medical line, the healthcare line, you start from a clinical problem, you analyze variation, and then you discover patient-specific patterns. So that's that. Are we still together, everyone? Are we still together? Are we still together? Okay. All right. Thank you. All right. So I will attend. I'm seeing some questions. I will attend to it. Let me run off with the slide presentation. And I will attend to your questions. All right. So that's case 3. Then okay, in neurogenomics, there's also something called neurogenomics. Maybe you're interested in genes involved in learning and memory. The research questions: which genes are active during memory formation? Which genes are active during memory formation? We are talking about gene activity now. And when you're talking about gene activity, that has to do with RNA sequencing. That's expression profiling. I can rephrase this: which genes are expressed during memory formation? So the data I need now is maybe RNA sequence from maybe the mouse of brain regions from post-learning. So a mouse that has been subjected to a learning activity, and then you sequence the RNA. That is after the learning activity and all of that. You now want to understand. You carry out QC, you carry out alignment, you carry out expression analysis, you carry out GO. GO, that's the gene ontology. That has to do with pathway analysis and all of that. And then you discover that BDNF and the CREB gene are involved in neural plasticity. and it was applied that the insights provided, it provided insight into Alzheimer and cognitive disorder. So disorders that has to do with memory. So this analysis will provide insight, So this was from a biological process. You make use of transcriptomics, and then you review functional pathways. That's that. Okay, why do people react differently to drug? I think we've talked about this already. That has to do with um all of this kind of data and all of that. You carry out variants and all of that. So that's just it. Then maybe you are even someone in the field of natural wellness, phytogenomics, medicinal plants. Even you can make use of bioinformatics in your field. And the research question can be that what genes produce essential oils in lemongrass? What genes are responsible for the production of essential oil in lemongrass? Then you carry out RNA-seq from the leaves and the flowers. You carry out assembly, you carry out annotation, and then you now discover that the terpene synthase genes identified in oil biosynthesis. So you discover that these terpene synthase genes are the ones that are responsible for oil biosynthesis. And the application can be that it can guide breeding for higher oil yield. So maybe you venture into oil production, maybe essential oil production and all of that, but you want to know in terms of maybe now using your plants. It will help to guide what that maybe, okay, the plants that can be used in production of these oils, we want to do what, breed them that these genes are well expressed. Or I can even turn this around, that at what stage, at what level, maybe in terms of especially those that deals with medicine, like natural medicine, maybe from plants and all of that, even giving a medicinal plant. Do you know that even harvest time, the timing of harvest even determine how potent such medicinal plants will end up to be? So maybe you are a herbal formulator, and then you want to know at what maybe is it at the early stage or at the late stage that this particular the particular benefit you want to get from that plant, at what stage is it highly potent? It will guide when you harvest your plants. So that's why bioinformatics is very interesting. And even specifically, we are genetures, we explore medicinal natural wellness. So that's the different things that we can get. Another case study has to do with diversity, maybe conservation. You want to preserving genetic diversity in wild yam. The research question: what is the genetic structure of wild yam populations across West Africa? When it comes to conservation, you carry out RAD sequencing from multiple regional populations. You carry out QC, you carry out SNP calling. SNP calling is that's variant analysis, and then you understand the population structure, the diversity analysis, and all of that. Then you can discover, you detected that high regional differentiation and reduced diversity in isolated population. So after all of this, your analysis and all of that, you are now able to identify that there is reduced diversity in a particular population. So what are you going to do? You apply it in it to now guide selection of genetically diverse accessions for conservation and crop improvement. So in this case, we started from a biodiversity concern. Can you see? This is all from the pipeline. It started from a question, and then you make use of genomics to review hidden population patterns, and then it informs your sustainable conservation and breeding strategies. I think there is a workshop that is even coming up specifically on this conservation genomics, where we are going to be exploring all of this, and you are going to be learning hands-on how you can identify whether a particular population needs to be conserved or not. I think that we are going to be starting that next week. Yes, we are going to. This was supposed to start this week, but due to some things, we had to postpone it till next week. So in that class, we are going to be learning this one even much more practically. So that, by the way, so I've been able to give diverse case studies which cut across, even if your field was not mentioned. Even from this one, I believe that you must have learned something. Even from now, you now start asking yourself, in that field you are, which problem can be solved? Which problem can be solved? Let me give a practical case study, even from Genomark Holdings as a whole. There was a research that was going on, because I believe we have heard about antimicrobial resistance, right? We've heard about antimicrobial resistance, right? Antimicrobial resistance. We've heard about that. So even at Genomark Genomark Institute, there was a research due to that was a problem, antimicrobial resistance. And then there was a project that was carried out titled ARES, that's antimicrobial resistance surveillance across African countries. So maybe, for example, it can be that you want to understand maybe, for example, tuberculosis, the how this the bacteria that causes tuberculosis, how it has developed resistance over time. Because if you are using old drug for something that has already developed resistance, you just be using drug and the thing will not work. So based on that problem, Genomark embarked on that surveillance system, tracking tracking antimicrobial resistance across African countries. That was a project of Genomark. So that was it. So I was saying that because I am talking about your respective fields, because a burden of Genomark is that even as we are doing programs, we are doing all of this, we really want to raise scientists, we really want to raise researchers that are solving real world problems. So as you are engaging in different workshops, as you are engaging in learning different tools, we really want you to make use of it in what? Answering real world problems, solving real world problems. And that's it. That's it. So I'm challenging us today. If you've not been paying attention to problems in your field, I'm challenging you today. Look for problems in your field, and then you can apply bioinformatics, whatever skills you might have learned, and all of that. Okay. All right. So once again, just emphasizing again, choosing the right pathway. Question will determine your choice, and then you see your expectation. So I'm going to share this slide if it to be helpful for you. So from the data to the discovery, you've identified the question, you've identified all of that. Now the right data type, DNA, RNA, protein, and then in bioinformatics, there are different bioinformatics platform databases where you can source for some of these data. Perhaps maybe you are carrying out sequencing yourself, or you are not carrying out that. Even if you don't carry out sequencing yourself, you can still make meaningful insights from available data that is already stored on different databases. Yes, I was talking about that surveillance project that Genomark was carrying out. How did we get our data? It was from the database, from different locations, from across African countries. So you source data. So two ways of getting data is that you carry out sequencing yourself, or you go to database. You can still make out meaningful research even from data that you get from database. So that's it. And then understand, then as a bioinformatician, you need to understand how sequencing data is generated and stored. The different sequencing platforms are Illumina, Nanopore, PacBio, and all of that. It comes in different formats. I think there are many bioinformatics workshops we've done that we explained all of these kind of formats, database, how to access them, and all of that. We have the FASTQ formats, we have BAM, we have VCF. There are so many formats. When we talk about formats, it's just like MP3, MP4. There are different formats, right? So even in bioinformatics, different biological data have different formats of storage. So perhaps you are exploring on the database. And I talked about, okay, use sequencing, even pushing research. That's the reason why some of these databases are available, that even if you sequence your sample yourself, you can upload it to these databases. And then for future research that other researchers can make use of, even you can go back to make use of it whenever you need, and all of that. So depending on the kind of data, you determine different kind of formats and all of that. And of course, the databases like the SRA, ENA, and so on and so forth. And then you make use of bioinformatics tools to extract meaningful patterns. When you talk about alignment, you carry out blast. Okay, I think I mentioned that we are going to be doing a little bit of blasting tomorrow. So prepare yourself. And then different bioinformatics tools, if it's for expression of variants, we make use of DSEC and this. Then visualization. I think I saw some people said they have skill in R, they have skill in Python and all of that. These are some of those visualization skills or softwares, tools that you make use of, Cytoscape, Mega, and so on. And then you translate results into insights and publications. So from whatever insights you get from your analysis, of course, you want to interpret them biologically through literature review, visual outputs like figures, heat maps, trees, that's like maybe evolutionary tree and all of that. And then reporting. You're reporting maybe in papers, in bioinformatics, and that's discovery for you. So in summary, bioinformatics is not just about the tools alone, it's about what? Connecting data to purpose. And that purpose stems from so you start with curiosity. Like I said, that's one of the core skills of a researcher, of a scientist, curiosity. And then you are guided with the data, and then analysis will reveal the insights, and discovery will create impact. And that's bioinformatics for you. So I'm done with my presentation for today. Tomorrow, we are going to be diving into the practicals. And I really want to hope that this session is able to even fulfill the desire even in our hearts for creating this. I want to believe so. So thank you everyone for following all through to this time. Thank you so much. So I will want to attend to some of those. Okay, in terms of uploading the slide, everyone on this platform, are you in the general platform in our community, WhatsApp community? That's where I'm going to be sending that. That's where I'm going to be sending the workflow. Okay, you are talking about workflow for tomorrow. Can you demonstrate workflow of all 7 examples? Sorry. If we want to do that, workflow of all 7, a whole day itself is not enough. Those things, they are deep. Those things are deep. Some of those workflows I mentioned, some people here might have even learned some of those tools and all of that. The link to the WhatsApp community. Mr. Ahmed, please, are you still on call? Can you help us to drop our WhatsApp community so that those that are not in the community can join? Those that are not in the community can join. I will be dropping that, and also to YouTube. I believe some people are joining from YouTube, so you can send the sample to, sorry, so that we can send the presentation to them. Or perhaps wherever you were able to join this session, you can just indicate. All right, so let me just attend to your questions now. Let me attend to your questions now. Okay, I want to make sure I'm not missing out on a question. Yes, for asking about the recorded video of this lecture, I think the is recorded, and even on our YouTube page is being live streamed. So anytime, at any point in time that you need to go over the session again, you can get it from our YouTube page. So if you are not following our YouTube page, on the group, you'll see that we've posted it. You can follow again, and you can always go back to rewatch the session on our YouTube page. Okay. Okay, this explanation for alignment, I thought that's genome assembly. Yes. I think at a particular point, you are right, Jonathan. Yes. Let me go over that again in case some people didn't get it. So when you talk about assembly, actually, alignment is also a form of assembly. Yes. Alignment is also a form of assembly. So if I say assembly, like you are assembling the different fragmented contigs together, yes, that's genome assembly. Genome alignment, that means maybe, for example, in a case whereby you are maybe for example, you want to maybe now, okay, let me come this way. I sequenced a plant sample, a particular plant sample, maybe a tomato plant today in this 2025, and I want to understand what this tomato plant of 2025 is. I need to align it to a reference sample. There's something called reference sample for plants. We have reference genome for tomato, we have reference genome for rice, and many more. So if I want to understand this one that I sequenced today, I will align it to the reference, so that when I align it to the reference, I'm going to, because when you are aligning, as the name implies, alignment, we want to be able to see, I want to be able to see the genes that are present in this my own, because the parts that are the same with that tomato plant, it will align with each other. Is also a method of assembling the genome. Is also a method of assembling the genome. Is a method of assembling the genome. Maybe when you want to carry out variant analysis, there's something we call mapping to reference. That mapping to reference is an alignment. Yes, mapping it to the reference genome is also a way of assembly, actually. There are different methods of assembly. Yes, so alignment is a method of assembly. Yeah, like that. So somebody said, what to do if plant is not abundantly sequenced, say for polyploid species like wheat? You see, this question you asked is one of the, especially in plants that are not popular. When I say plants that are not popular, I think much research has been done on maybe global crops like maybe rice, tomato, and all of that. Then when you now talk about maybe, especially for we in the maybe medicinal aspect, we discovered that maybe on databases we don't have sufficient data compared to plants like the rice and the co. So what to do? That question, that's why we are coming up as researchers, that's why we are coming up as scientists, that we can carry out these sequences by ourselves. And that's another thing, that bioinformatics is not, I would say, is a research that is a kind of thing that is it's a collaboration. I think, okay, there was one workshop I also presented that was talking about bioinformatics that has to do with collaboration and all of that. Maybe a research project in school. Of course, if there are no sequence plants, then they need to be sequenced. So that's just it. Then where only one variety Chinese spring is fully sequenced and no other variety in the entire world. Okay, no other variety in the entire world. Anyway, yes, only one variety is sequenced, just as you have said it. If only one variety has been sequenced, and maybe in a location, I will still put it to your research question. If you are someone that is interested in answering that, and you have discovered that only one variety has been sequenced, and there are some other varieties that are all over the places, or if I even want to streamline it to your own specific location, then it's now up to you to do what? Sequence from your location or anything, because it starts from somewhere. It starts from somewhere. It starts from somewhere. It starts from somewhere. So that's just it. Somebody said, will it be possible to sequence the genome of every individual, as we always preach on personalized medicine? That's that. To sequence of every individual. You see, precision medicine. Wow. Maybe I think we need to share a lot of things, though. A lot of things needs to be shared. Maybe I'll go and look for one of the YouTube presentation of this precision medicine and send to you. And it was a speaker from India that was talking about it, how that has been possible, because even that person, she has a precision a genetic lab for testing and all of that, how that has been possible. But let me just answer in the little way I can currently, that will it be possible to sequence genome of every individual? You know, in the case of, maybe in the case of cancer now, for example, in the case of cancer, maybe someone has been using drug, and then it has not been working for the person and is working for another person. Of course, in that case, you will know that you need to sequence for that person, right? You need to carry out genetic testing. Yes, that's the word I want to use, genetic testing. We used to do a lot of different kind of testing, but we are lacking in genetic testing in Nigeria. We are lacking in genetic testing. So even genetic testing has to be prescribed, just the same way you prescribe lab tests for people. Is the same way you can prescribe a genetic testing when the situation arises. So it's just like every other normal test, so to say. But in this case now, we are dealing with DNA profiling. So for a person that needs it, of course, it can be sequenced. It can be sequenced. It can be sequenced. It can be sequenced. That's why sometimes some of these things, they'll say they want to fly this person abroad because the the tools necessary is not available and all of that. So we are coming there. Let me just say we are coming there in Nigeria. We are coming there. Okay. Where in Nigeria can we carry out sequencing for insect genomics? Insects. Where in Nigeria? Okay, I don't have idea of insect genomics, but of course we can always search that out. Okay, Mrs. Gloria, let me say I can answer your question in the other class. Let me put it there. I will answer your question in the other class. Yes. But if you have idea of some of these sequencing platforms, sorry, places that they carry out sequencing in Nigeria, you can make inquiry from there, that can they carry out sequencing for insects? Can they carry out sequencing for insects? So it's just a matter of we making research on it and we'll be able to track it down. So that's just it. So Okay. From the first case study, which is on rice, you mentioned sequencing RNA for drought tolerance. Okay, why not DNA? Why not DNA? That I mentioned RNA, and why not and why not DNA? I also said it. I think when I was explaining that, I also said something that even from this case study, I can still put it in different ways. That was when I was talking about the fact that if it's DNA, yes, it's still in that part. Okay, maybe I should go back to that case study. Maybe I should go back to that case study. Let me go back to that case study for rice. Okay, did you say? Let me look at your question again. Sorry. From the first case study, sequencing RNA for drought tolerance, I think based on what is on the slide currently, I think I will have to reshare then. Whole genome sequencing, whole genome, that's actually a DNA sequence. That's a DNA sequence. That's a DNA sequence. I think you are talking about the first case study. First case study. Whole genome resequencing, that's WGS. That's WGS. Is actually that's DNA sequencing actually. That's DNA sequencing. DNA sequencing. And even beyond it, even beyond DNA sequencing, if I want to progress to RNA, that okay, I've been able to identify this gene. That's identification of a gene. That's from the whole genome sequencing. That's from the DNA. I've been able to identify that this gene is present in this one and not in the other one. But if I want to go beyond just identification of gene, I might want to get the expression, how it is being expressed, That's where I now enter RNA sequencing, actually. So DNA is like you knowing what is present. RNA is like you knowing that thing that is present. How is it expressed? So that's it. I don't know if I answered your question. So someone said, should I use the same Genius tool used to do mutation annotation to do predicting drug discovery and personalized medicine for that mutated gene sequences? Yes, in Genius tool, Genius software has different pipelines. If you remember when you were installing that Genius software, you carried out, you installed different plugins. In that tool, there are tools that you make use of in that same Genius software to actually predict for that. They still use that same tool even in drug discovery. So it's just for you to know, maybe it's for you to know how do you actually make use of that Genius software to do drug discovery. So that's not something for this class actually. So if you engage in a drug discovery class, you'll be able to see how that same Genius software can be used, or you can even exploit yourself to see how you can make use of the Genius software to do to carry out analysis for drug discovery and all of that. Okay, YouTube question. Is it possible to do bioinformatics on yam sample? Of course, you can do bioinformatics on anything. Depending on what insight do you want to derive from the yam sample, that will now determine, you now sequence the sample. So just like we have been talking about so far, I think I even gave one. Okay, the example of yam I gave was on conservation. So your own question might not be a conservation question, it might be another thing. Are you trying to find how it's tolerating a particular stress condition? Then you sequence, you carry out sequencing, and bioinformatics has started like that. Okay. As in last question, drought and ABA response in rice. Can you please present detailed workflow with project proposal? Wow, using bioinformatics tools. Can we do that in this class? I don't think so. That's where you are talking of maybe like preparing a research proposal, abi, detail case study and all of that. Since if you can remind me tomorrow, we can do that after the practicals we have prepared for tomorrow. If we can chip that in, we will. If we can chip that in, we will. Okay. So you said you have BioPython crash course tomorrow. No problem. If you miss out on tomorrow class, tomorrow's time, can it be adjusted? I don't think the time cannot be adjusted. At best, you can always go back to the recording and it will still be streamed on YouTube. So you can always go back to it whenever you need it. So someone said, now I'm seeing that this bioinformatics cuts deep. The name only seems simple. It can also cut deep into research of the drugs that doctors use. Yes, it's cut across that doctors use. Maybe you can even carry out maybe molecular docking, maybe like protein drug interaction, maybe how drug binds to proteins in the body. So that's a drug interaction. You want to understand the interaction of drug with so you want to do this drug interaction that has to do with the proteomics field, all of those molecular docking and all of those things, understanding drug interaction and all of that. Okay, so asking for LinkedIn. Yes, we'll resend all of that to the platform. We'll send all of that to the platform. We'll send all of that to the platform, following all our platforms and all of that for you to follow. So that's that. I think I've been able to go through all of the questions, except you have anything for me again. We're already coming to the end. And okay, it's very important that I'll still like one or two to unmute their mics and maybe give a comment on today's session. But before I do that, in preparation for tomorrow's class, we are going to be carrying out a hands-on practical session. Yes, of course, we need a tool. We need a bioinformatics tool for that. We need a bioinformatics tool for that to carry it out. So some people here already have Genius software. That might have been from maybe previous workshops that you've done, that you had the software, you're able to have the software, and it's still the same software that we're going to be making use of. So if you are here and you already have Genius software, this is not applicable to you. But for those that don't have Genius software, yes, the software we're going to be making use of tomorrow is Genius software. So if you don't have the software, you need to have the software installed before tomorrow. So there are two options. That software is actually is a software that you need subscription, and the price is very high. We are talking about $500. We are talking about $1,000 and all of that. So that's what the tool is about. But there's a free version actually. There is a free version. Certificates. Wow. I don't. Okay. Let me answer that question tomorrow. I will answer your question tomorrow. Although we didn't plan certificates for this training. It wasn't planned for this training. It wasn't planned for this training. So as I was talking about the software, there is the free version. There's the free version. Of course, it's a 30 days trial. So if you don't have the software, you can make use of the free version. But if you wish to have the software, that you don't want the free version, we can give you the lifetime activation key, for it. But that will come at a cost. Actually, it will come at a cost. it will come at a cost. So very quickly, let me know if you have the two already. If you have Genius Software already, just type yes. And if you don't have Genius Software, type no. So we'll know the we are going to know the amount of people that don't have. Okay, I've seen one yes, I've seen one no. No, 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 no. Mr. Ahmed, please, are you still on call? Hello, Mr. Ahmed, are you still on call?
Yes, yes.
Okay, all right. So now a link will be dropped now. A WhatsApp link will be dropped now. But you are only going to join that WhatsApp group if you need the lifetime activation key. It's going to come at a cost, and is a very low cost. When I mean low cost, in fact, it's not a cost, but it's just, you know, it's just for administration. It's just for administration. That cost is just for administration. Just like I said, the price of Genius Software is, maybe I should show us. Genius Software price. Genius Software price. Now, just for students alone, can you see that $200 for students alone? The subscription for students is $200, and so this is it. Okay, I believe you can see my screen, right? Okay, where is it again? So for personal, $9,500. For corporate, for academic, $4,750. That's it. So this is there was a place that was showing this thing for students and all of that. So you've seen a little about what the price. So if I call a price currently, is that it is at a very ridiculous price, but of course we want everybody to be able to get it. So for those people that already have, that's, you've gotten it from our previous workshops that we've done and all of that, you've been able to get it. All right, so now the offer currently is that if you want to just have if you want to have no, there's no web version. You have to install the app. So a WhatsApp group will be dropped. If you want to get the lifetime activation key, you'll be getting it for just $3. For just $3, lifetime activation key. So that the WhatsApp group has been dropped. So if this is for, if you want to get the lifetime activation key, please don't join the group if you have it already or you don't want to get. If you don't want to get the lifetime activation key, we are going to show you how to the free installation that you can use in preparation for your next class. The free installation that you're going to use in preparation for tomorrow's class. So we are going to share with you how to activate the free version. Or should I say that if I say those that don't have it at all, everybody should join the group. I don't know what's the best approach now. At first, I just wanted those that will want to pay $3 to get the activation key to join this group. All right, maybe what we are going to do, yes, because we are going the purpose of the group has been achieved. So it's just for you to get the activation key and then you exit the group. So we are going for those that are not going to get to pay to get the free the lifetime activation key. we are going to share with you how you can install the free version. The free version, of course, the free version is for 30 days. And like I was saying before, is a tool that can carry out is a software that has different tools that can carry out different analysis. So the free version is limited to what it can do, but it will be so okay, those that want to get the software, yes. So those, we are going to stick with the way it is. Those that want to get the software should join the group. But if you want to get the if you want to get the, so Mr. Ahmed, please, you can still drop the, oh, really? I was even saying 30 days. The free version is just for 14 days. The free version is for 14 days. So please, you can help us to paste the link here of how they can. Please, everybody should go through the video and prepare and have the tool so that you can be carrying out the practical as we are doing live. We dropped now for you to be able to have the free version installed. So you can drop the free version here. So those that can get it now can get it, and we might also resend it on the group if you didn't miss out. So please, before we end this call, this is the link, this YouTube link will guide you on how to. Okay, the lifetime activation key is $3. Is $3 for Nigerian payments. Nigerian payments will just be I think that should be 5,000 naira. 5,000 naira. No, you cannot install the app on your phone. It's an app for laptop. You can't install it on your phone. You can't install it on your phone. You can't install it on your phone. So please, you can get this link now. Maybe if you open the link here now, you can maybe bookmark it or something so that you can, please let everybody have the software installed before tomorrow. Before tomorrow, same time, same Google Meet link. I think I'm done today. I'm done today already. So I'm done today, and we are going to be ending. We are going to be ending now. So please make sure that you've gotten the the YouTube link that will guide you on how to install the activation. And those that want to join to get the page the page that want to pay for it, so you can join the group. I'm going to give 5 minutes for us to settle all of that, and then we are going to be ending the call now. We are going to be ending the call now. And one other thing, as we end the call, please, I would like you to share your feedback on our platform. You can share your feedback of today's workshop on our platform. That's our WhatsApp general community. And you can also even share it on your respective handles, tagging us, so that if you know that there's somebody that still wants to partake of tomorrow's free session, you can talk about today's first session. Talk about it on your platform, tag GNatures, share the registration link to them, so that tomorrow we can all partake of it. So I believe is a good point to pause. It's a good point to pause. Thank you, everyone. It's really a wonderful one that you were able to stay all through till the end. Thank you so much. Till we meet tomorrow. Let's not forget to give our feedback in the group chat and everywhere that you can give your feedback. Thank you so much. Thank you so much. So we can exit the call now. Thank you, everyone. Goodbye. Thank you. Thank you.